Hello everyone, we are from Group 5 of Medical Biotechnology. We'll be presenting our video titled Lung Cancer, the Silent Killer. And this is our group members.
Introduction to lung cancer. Cancer is a disease in which the body cells proliferate uncontrollably, where the DNA of normal cell will be damaged and turns to defective cell. After undergo cell multiplication, the cell cancer will be formed. Lung cancer starts in the lungs and can spread to lymph nodes and the other organs. The disease has no specific symptoms or signs that allows for early detection. Due to this, most of the patients of lung cancer will present with advanced stage. Lung cancer consists of two main groups, which is small cell lung cancer and non-small cell lung cancer. There are 15 to 20% people diagnosed with small cell lung cancer. It is usually caused by smoking and the cancer tends to spread quite early. Small cell lung cancer also classed as neuroendocrine tumors. There are three main types of non-small cell lung cancer, which are adenocarcinoma, squamous cell carcinoma, and large cell carcinoma. These three are classified as in the same group due to their behavioral and response to treatment being similar. Around 80 to 85% in the UK diagnosed with this type of lung cancer. Statistics in Malaysia shows that lung cancer is the most common with approximately 10% of all malignancies.
Now, let's discuss biological pathway of lung cancer. Basically, there are two types of biological pathways involved in lung cancer, which is known as metabolic pathway and signaling pathway. Signaling pathway involved is known as RAS pathway, which is a pathway regulated by RAS gene of the lung cancer. Metabolic pathway of lung cancer. In metabolic pathway, malignant lung tumor cells tend to yield ATP via lactate fermentation or in the presence of adequate oxygen. During fermentation, lung cancer cells tend to utilize large amount of glucose. As a result, high glycolytic rate and overexpression of hexokinase isoform enzymes takes place. Whereas in anaerobic fermentation, glucose consumed meant for generating large amount of lactate, which eventually being excreted by MCT4. MCT4 stands for monocarboxylate transporter 4. As a result, it will induce the Warburg effect that prefers the formation of lactate instead of forming acetyl coenzyme A. Extracellular acidification, promoting excess of lactate from MCT4 transporter, which contributes to invasion and metastasis of tumor cells. Now, let's move on to signaling pathway. In signaling pathway, RAS gene that encodes a G protein is essential in signal transduction pathways, which control cell specialization, proliferation, and viability. It works by binding to GDP, which is inactive in quiescent cells. GDP is activated by stimulating the upstream growth factor receptors, which leads to the formation of GTP. RAS GTP that are in active state then binds and activates subsequent downstream pathway, including RAS, RAF, MEK, PI3K, and mitogen-activated protein kinase (MAPK) pathways. RAS is necessary for downstream signaling induced by a diverse array of transcription factors, including epidermal growth factor receptor (EGFR), and constituent protein stimulation eliminates the necessity for growth factor facilitated signaling. Activating mutations modify the protein's GTPase activity, restricting inhibitory effect of the active RAS GTP to GDP, resulting in greater signaling pathway via numerous downstream growth promoting pathways. The RAS, RAF, MEK, MAPK signal transduction cascade is required in several lung cancers and consists of approximately one mutated gene.
The adaptation of biotechnology in treating lung cancers can be seen by the utilization of immunotherapy in fighting the cancer cells. Immunotherapy is used to activate the auto-defense mechanism of the body by treating the patient with exogenous substance, or also known as immune checkpoint inhibitor. Immunotherapy is crucial to regulate the immune response to tumor, as well as helps in stimulating the immune cells to identify, inhibit, and kill the tumor cells. The checkpoint inhibitor is a type of drug that's responsible to block the protein from binding on immune system cells, which then allow them to fight tumor cells. Semiflimab and durvalumab are examples of inhibitors that are available in the market. In specific, the inhibitor responsible to increase the strength of anti-cancer immune response by targeting the proteins PD-L1 and PD-1. When there is no binding between PD-L1 and PD-1, the T lymphocytes will be stimulated and the tumor cells will be killed easily. Thus, the abnormal upregulation of tumor cells will be prevented.
That's all for our video presentation. Thank you for listening.